welcome to all my colleagues to this small presentation on MSCs, a little bit on the background, on the biology, but then mainly about clinical data that is out there for MSCs and the pediatric and adult setting in patients with steroid refractory acute GvHD. So here you see a proposed mechanism of action. It says proposed because none of these mechanisms of action here has been shown to be the only one, and the other ones have, so to say, not been ruled out. It is known that MSCs, when you transfer them, and patients, even if they respond, it's very hard to find and trace them back after transfer. But it is shown in vitro that these MSCs release so-called extracellular vesicles, EVs, and these EVs contain RNA or other immunosuppressive molecules. And this RNA can enter the target cells and change transcription or be transcribed itself. These vesicles can also contain proteins, peptides, hormones, and immunosuppressive molecules like, for example, kynurenine, which is generated by an enzyme called indoldiamine deoxygenase. And this enzyme IDO generates immunosuppressive kynurenine and leads to the metabolism of tryptophan, which is then lacking in the microenvironment and which is important for activated T cells. And thereby, also by the transfer of organelles via tunneling nanotubes, the target cell may be influenced. There is some evidence that even regenerative effects are exerted by MSCs. Also here the mechanism is less clear. What is clear and what has been shown uniformly is that MSCs generate immunosuppressive kynurenine, immunosuppressive adenosine via the ectoenzyme CD73, and that they lack HLA class II expression, so they can be administered to patients even if they are third party, because they will not be recognized because of their lack of MHC class II without donor-recipient matching. This is one case report, one of the first cases actually reported by Katharina LeBlanc in 2004. It was a child, 9-year-old, and this patient had grade 4 acute GvHD and was refractory to steroids and several other second-line therapies, and was then treated as an individual treatment approach with bone marrow-derived MSCs from the related donor. Actually, the first infusion already improved the condition of the patient, GvHD improved, but after the second infusion, the patient achieved a complete recovery and complete remission of the acute GvHD. This was published at the time, I think, in Lancet as a case report, and was the first really clinical proof that this cell population has clinical activity against acute GvHD, and was, so to say, the starting point for several clinical trials. Some of them I will highlight in the coming slides. Yeah, this was a study for patients with severe steroid refractory acute GvHD treated with MSCs within the EBMT ex vivo expansion procedure in a multi-center phase 2 trial that was then led by Katharina LeBlanc and published in 2008. And what you see is that when you look at the overall response rate, 39 out of 55 patients responded. This is a majority. And she found that there is a correlation between responders versus non-responders. This is not unexpected. Responders had a better overall survival at 2 years after transplant, with 53 versus 16%. And survival in general was poor, with only 21% patients out of 55 surviving at the time point of 2 years. This was a trial that led to approval. Remestemcel-L was tested in a phase 3 but single-arm multi-center trial and compared to a so-called pre-specified control. overall response at day 28 was the primary endpoint, and remestemcel-L induced in 70% a response versus 45% in the control. Steroids could be tapered. The drug or the stem cells were generally well tolerated. Main complications were infections, GI disorders, general disorders, and so on, which is not unusual in this pre-treated patient population that had received corticosteroids before. And based on those data published in Biology of Blood and Marrow Transplantation 2020, the FDA approved remestemcel-L for steroid-refractory acute GvHD in pediatric patients in 2024. This is another stem cell preparation that was tested in Japan, actually, JR031. Was tested in a multi-center phase 1/2 trial in patients with grade 2 or 3 steroid-refractory GvHD. By week 4, 13 patients responded to the therapy. You can see here complete remissions 8, partial remissions only 5. The follow-up phase 2/3 trial in patients with more severe graft-versus-host disease, grade 3 or 4, showed out of 25 patients durable complete remission by 12 patients. And survival of patients that had an overall response was superior to those who had no response. Not unexpected, patients that are responding have a better chance of survival. This trial led to approval in Japan by the Japanese Medical Device Agency for the treatment of GvHD in children and adults based on this trial in 2015. Yeah, there's one problem when you look at all these clinical trials or retrospective analysis with MSCs is that the product varies a lot. Depending on which trial you look at, the generation of the product is a little bit different. The control criteria for the products are a little bit different, and therefore also the responses are different. So the question is, how can this be made more uniform, and how can we have a safe product, ideally from multiple donors? And this was solved by a preparation called MSC-FFM. So it was from Frankfurt, a pooled MSC product from 8 different HLA disparate donors. This preparation was then tested in patients that had steroid-refractory MSCs. It was allowed to give that based on the hospital exemption clause of the European ATMP guidelines, and a metadata analysis of these patients that received this MSC preparation showed at day 28 overall response rate of 80%, and the differences in results may be due to highly variable MSCs products. Anyways, this was a good signal for efficacy, leading to the following prospective trials. Here, these are the real-world cohorts, outcome of patients with steroid-refractory or treatment-refractory GvHD in different countries. Here, day 28 responses, CR in 22 patients, PR in 35 patients, very few non-responders. Overall safety good, only nausea, vomiting, and headache, which is relatively mild. Again, responders were more frequent, 55% responders, and the patients were heavily pretreated, including ruxolitinib, and had a median survival of 4.8 months. A very safe product overall, only 5 temporary non-serious mild adverse events. Yeah, now we come to the 2 randomized trials that are ongoing. The first trial that I would like to introduce is the BALDA trial, a randomized open-label multi-center phase 2 trial. Here we have pediatric patients with steroid-refractory acute GvHD that receive these MSC preparation. MSC-FFM, overall response at day 28 is a primary endpoint. These are the secondary endpoints that are indicated here. And then there is an adult trial, the EDUN trial. It's a randomized open-label multi-center phase 3 trial. Patients are adult and adolescent patients that have, per definition, step-refractory acute GvHD. Primary endpoints are overall response at day 28 and overall survival up to month 24. This trial has recruited, I think, 2/3 already of the patients. So what we should discuss today is how could MSCs address current unmet medical needs in acute GvHD treatment. I think there's a lot of room for improvement. I think we have made improvements over the last years with different drugs, in particular with ruxolitinib, that is approved in the US and in Europe for step-refractory acute GvHD. But clearly there are many non-responders or patients that lose their response. And the question is, how does MSC compare and how can they add value either in combination or in rux-refractory patients? And then the next question is, in your experience, which patients would benefit most from this MSC therapy? How might the data from the two prospective trials that I just introduced to you impact the clinical landscape? Of course, this depends on the outcome of those trials, the results. And the fourth question is, what might be the key clinical considerations for clinical practice when treating patients with acute GvHD that receive MSCs? So with that, I would like to open the discussion, and maybe we could start with the first point on how could MSCs address the unmet medical needs in the GvHD field. Maybe we can start by saying what are, in your opinion, the unmet needs in acute GvHD treatment.
So thank you, Robert, for really a beautiful overview and summary of state of the art. Just if you allow me, before this, maybe the question which is puzzling for me, and you explained the problem about lack of standardization. How do we define today a mesenchymal cell? Because all of these trials use mesenchymal cells, but they are different products. All of them are allogenic, which is fine. But how do we phenotypically, for instance, define a mesenchymal cell? And my second remark is we're very fortunate having now these ongoing randomized trials, but how do we consider and what are the learning we had from the failed phase 3 randomized trial from 2009? You may remember the Osiris prochymal something trial, which was a phase 3 randomized trial, and it failed. So I think this context is important to move forward for the future.
Yes, I absolutely agree with you. So for MSCs, there are defined markers like PDGF-alpha receptor positive, SCA1 positive, CD73 positive, and there's a functional characterization that they can differentiate into cartilage tissue or bone tissue, depending if you go add certain growth factors. So that is something that has been done. And ideally for GvHD treatment, they should suppress T-cell responses in vitro. So you can do so-called mixed lymphocyte reactions where you can show that they are actually immunosuppressive. All having said that, so these are kind of quality criteria, but I fully agree with you. It was very heterogeneous, I think, before. Now I think these two trials, they have a very well studied, very well defined cell product pooled from different donors that has been characterized multiple times, including in vitro studies. That is relatively well characterized, but still it's not the same as giving a drug that inhibits a certain pathway, right? It's certainly still, as a cell product, always a bit heterogeneous. Yeah, and that, of course, makes it more difficult than giving just a kinase inhibitor, for example.
So if you allow me another question, unless my colleagues want to jump in, and maybe our pediatrics colleagues, Dr. Harris, can join us. Welcome to the hub, Andrew. Is about it has been always, and beside the approval of MSCs in the US for in pediatrics, but when we look even to Katarina Leblanc data from 2004, 2005, without any p-value, the numbers were always better in the children. Is there something specific to pediatric GvHD to be more responsive and to mesenchymal cells compared to adult? This has been always puzzling for me immunologically.
I think that's something that we all certainly see in our patients, and I think it's been fairly consistent in trials that children do tend to respond better to GvHD therapy. I think there's a few things that can be looked at here. I think one of them is that our children are much more likely to have good thymic function, so there's a better chance for positive negative selection once you eradicate these clones of T cells that are pathologic for our GvHD process. So I think that's part of it with our children. I think that also, with the lack of aging for our kids, there's a lot less damage-associated molecular patterns and things like that as well that I think also are part of the prime for GvHD. So when you get through this initial wave of alloreactivity, I think that our children are just much more primed for establishing better immune tolerance. One thing we've seen in this in the MSC trials, I think across the board, certainly is the safety of these cells. So I think that's one thing that is a very favorable feature of this treatment, is that it does not seem to increase risk for infection, does not seem to increase risk for relapse. So this is something that I think of more along the lines of ECP as far as safety goes, that I can use in patients that have a lot of other complications. With the product that is available here in the States, remestemcel-L, there was a retrospective look through John Levine and Jamie Farrar through the MAGIC Consortium that also showed that Ann Arbor 3, so unfavorable biomarker-defined GvHD, remestemcel-L was the first product in a retrospective analysis that showed some potential survival benefit for these patients with really unfavorable biomarkers. That's something that needs to be looked at and validated in a prospective study, but something that certainly gives reason to consider these cells in some of our highest risk patients, at least from a biomarker standpoint as well.
Could I clarify something? Andy, would you say that, how are you, Andy? Would you say that that the pediatric, that just kids respond better to any GvHD therapy, right? It's not just It's not just an MSC signal, it's just any therapy we study, kids will respond better than adults, correct?
Correct. That's exactly what I was saying even. Yeah. Okay. Yeah. And the other thing that is a challenge here in the States, and something that the pediatric community has been very vocal about, is just the sheer financial toxicity of the product that's approved here in the States. You're looking at, for a course of therapy, anywhere from $1.6 to $2.4 million for these cells that are third-party, off-the-shelf mesenchymal stromal cells, which is something that we certainly have to consider even here in the States.
How does the product being studied in these two prospective trials differ from what's available to us in the United States?
So I can only speak for the EDUN trial, and there the BAT. There's a list of BATs. Mainly ruxolitinib is used in the BAT arm, but also ECP, or it's also allowed, I think, to use MMF and mTOR inhibitors, for example. But most patients.
Sorry to interrupt you. My question really was, what's the difference between the MSC product and remestemcel-L? But since you brought it up, my second question will be, is that the right design, or should it all be ruxolitinib in second line? But
yes. Well, I can only talk about the product from Frankfurt. I think this is a product pooled from 8 different donors. I don't know how remestemcel-L is generated. I would have to look that up. But it's a different product, definitely. It's a multi-donor product in case of the Frankfurt derived MSCs. I don't know if they've been characterized against each other. I don't know any data on that, I must say. And to your second question, at the time when the trial started, I think Ruxo was not approved yet, or was just about to be approved in Europe. And therefore, they had to allow also other therapies in the BAT arm. But Ruxo is the most frequently used therapy, and there's a few patients that were either not eligible for Ruxo for certain reasons, and they received ECP, for example.
So we have Dr. Wolff, Dan.
Yeah, thanks for the discussion. To me, the discussion kind of reflects the challenge we now have in chronic GvHD disease, with a couple of agents being FDA approved. And now so what would be the optimal treatment? So PharmaMart has now makes the fecal transplant available. We have those trial running, or it was running for GLP-2 agonists. So, and the question, where is the MSC located in? That's something which may be more answered by logistics, financial aspects, and I haven't identified anything which would predict, as in chronic GvHD disease, to be honest. This is the ideal patient for MSCs. We agree that, or no, we don't agree, but there's no strong indicator that MSCs increase the risk for infections. There is some data, but that's probably the population we treat. But having an idea, and that's, I think, the biggest research need, where exactly would be the population taking benefit from MSC would be my most important need. And until then, it's just one of those agents you could consider third line, unless it's proven in second line superior to ruxolitinib.
I think Eben has a question or comment.
Yeah.
Is that right? You have your hand raised?
Yeah. Yes. I would also add that in the U.S. here, at least, we've struggled with what Dan was saying there. I mean, we've struggled to figure out how to conduct subsequent trials in either second line or third line, with the diminishing population and also the various exclusion criteria of the patients we actually see in these lines, and then the actual feasibility of these trials. But we should mention that in the U.S., we will probably along this topic, we will probably have a phase 3 trial in the U.S. using remestemcel-L being studied in the second line. It'll probably be a phase 3 double-blinded Rux versus Rux plus remestemcel for second line. And that was in the large discussions in the CTN, that was the best compromise we could find of actually being able to do a trial. I think many of us were worried about doing a large phase 3 in the third line in terms of various feasibility issues. And it just illustrates the challenges of the trials in this population.
So I'm glad you're mentioning this, Eben, because here you're raising actually 2 points. One point, and that can answer, I think, Robert's first question, where clearly resistance to Ruxo or absence of response to Ruxo is becoming more and more the biggest unmet need. And this is where Dan mentioned, beside MSCs, FMT, but also some other small molecules being developed. But also what you are raising, and this is why actually the ongoing phase 3 or planned phase 3 trial are going into earlier lines of therapy, is that it's becoming almost impossible to run a randomized phase 3 trial in this third line and beyond, because I'm always struggling with what should be the control arm, because if they have failed before, what would be my best control arm? Nothing is approved, nothing is proven effective. And obviously the numbers will never allow to achieve a full phase 3 randomized control trial. Even if you do it in, I don't know, 50 centers, in a reasonable amount of time. I don't know what are the thoughts of and this is why the trial planned in the US is moving to combine with Ruxo.
Yeah, randomizing third-line acute GvHD disease steroid refractory is just not a clever idea.
Yeah, but it's not a clever idea, but the regulatory people, Dan.
Yeah.
It's not. We agree with you, but it's what the FDA insists on at this point in time. That's the problem. So if it's an agent that you need regulatory approval for from the FDA for people to have access to and to use, unfortunately, we're stuck doing a randomized trial of some sort, and we agree, but that's where we are.
So then you said it's not a clever idea. What would be the scientific argument where one can provide to counter this?
Yes. So you're asking to reply to FDA. So I like actually the approach, and Robert, you did the same with the GLP-2 agonist add-on. And say, okay, I go for Rux and add something on which doesn't add to toxicity and look whether the response rate goes up. The trouble with a third-line setting is that it's usually steroid refractory, severe, grade 3 and 4. The patients will die whatever we do in a significant proportion of patients. And that's something which I would strongly avoid, and because it's just not a pleasant trial, and picking for the clinical use, picking up patients way earlier and improve their prognosis would make a whole lot of sense. That doesn't mean that randomized trials are not important.
Yes, but if I understood even correctly, this is what they are doing, right? In the US, they are combining Ruxo plus MSCs versus Ruxo alone, right? And this,
I think we've acknowledged that Rux, while a great drug, and in addition, for a lot of reasons, for the steroid refractory, especially lower GI disease, is not optimal or satisfactory by any stretch, right? So we still think there's room to improve in that high risk steroid refractory population on top of Rux, but with the field's embrace of Rux, it just it becomes very difficult for the various reasons already mentioned to do a third-line trial, to get that third-line patient on, and then the various competing risks of that third-line population to actually see something work.
So then I'll jump in again to help Robert move forward with his questions, because you then also mentioned, and would love to hear from everybody about, assuming today you have the MSC trial, you have an FMT trial, we have the GLP whatever trial, and maybe another drug. Do we have any clue where to orient or direct a given patient? For instance, a patient with maybe severe exclusively or majority of GI acute GvHD would rather go to this treatment versus another treatment. If it is more in the liver, I don't know, although this is not the most frequent one, we should go to something else. Any thoughts on this, especially from the colleagues who used a lot of MSCs, if there is a phenotype of GvHD that respond better?
I would just argue that would be a lovely problem to have, having multiple agents that we think are actually effective in the second line. So I think it's premature to have that discussion.
No, Corey, my question is about today. You have 3 trials for Ruxo refractory acute GvHD. What would you do?
We would not have 3 trials.
Correct.
We don't have enough people for 3 trials, but if we had 3 trials, I don't know.
Finish one, move on. Prioritize the one that's closest to finishing. Prioritize the one that's phase 3. I mean, I don't think we can say anything.
But I think the question makes sense, because in the end, we want to give our patients the ideal treatment, and we will learn from these trials that are ongoing what might be the best treatment, right? We can do sub-analysis and then see if skin responds better to MSCs versus GI tract and so on. There are some clinical impressions that we get already. I've seen a couple of severe acute skin GvHDs responding very well to MSCs. If that is something that is a signal in the end, we will see, because I oversee only the patients in Freiburg. But if that, so we will learn from those trials and see if there's a certain type of acute GvHD that responds best to MSCs versus ruxolitinib.
And actually, Robert, this is interesting because you are reminding me now that in the 2009 failed randomized trial, there was a better response and subgroup analysis for liver GvHD. And even I think at some point the company wanted to get approval in this specific niche. I don't know what was the end of the story, but I think it's not going to be the same story. I mean, we all agree that GvHD is very heterogeneous, whether acute or chronic. So it is unlikely to consider that one drug or one agent will solve all organs and all features of severe GvHD.
Yes, absolutely. Yeah. Maybe we should continue with the questions that we have. So the next point of discussion is how might the data from the phase 2 BALDA and the phase 3 EDUN trials impact the clinical trial, the clinical landscape, right? And this, of course, depends heavily on the outcome. If it shows that it is equally good, for example, MSCs to ruxolitinib, it might be a good combination partner because the mechanism of action is probably very different from ruxolitinib. If it's significantly better than ruxo, then of course it could become the standard second line. If it's significantly worse, then probably we keep on giving ruxolitinib in second line and MSCs, depending on the response rate, then in third line. These are the potential outcomes. But this is, of course, something we can discuss.
Robert, I'm not familiar with these two trials. I know they are running in some centers in France. Is there any interim analysis being planned? Because obviously one would like to avoid including 300 plus patients before knowing the final answer. Where do we stand with these trials?
Yes, the interim analysis was already done, I think in February and March this year, and showed that there's still a chance for achieving the primary endpoint. So it continued, and there were no safety signals. So I don't know the data. No, it's blinded, obviously. We don't know. It's not official, so to say, but it was good enough apparently, so that the trial continued.
At least there's no safety signal.
There's no safety signal, and there was no signal that the primary endpoint could not be reached.
Okay, so these are just I'm ignorant of these trials as well, but just to clarify, obviously we don't have the numbers listed here, but in terms of the total N, but I'm assuming these are both designed for superiority for the primary endpoint, and they're not big enough to have a non-inferiority endpoint here, right? So for MSCs to succeed in both of these trials, they have to better the control here, right? And is the phase 2 randomized to BAT, and both are randomized to BAT, or is the phase?
Yes.
Yeah, both are randomized to BAT. So if the majority use Rux, I guess, or Ruxo, as the Europeans say, I will say that so the MSCs have to do better than Ruxo here to have a chance at being approved. Is that or you guys already have access to these commercially? Like, are these being done for regulatory approval, I guess is the question.
Yes, this is done for regulatory approval. And there is this exemption program in at least in Germany, so that you can purchase MSCs. These MSC-FFM can be purchased. But this approval was conditional to doing a phase 3 trial. and therefore this trial is running now. And but independent of that, these MSCs can be purchased.
So
Does
to
the ability
answer,
to purchase them go away if the trial is negative?
Yes, probably they will take away this approval. Yes.
Yeah. Well, in theory, that is, Corey, the concept of the conditional approval, that if the confirmatory trial is negative, although I think your point is well taken, we've seen situations where the confirmatory trial didn't make it until the end, but still, given the efficacy, safety, and widespread use of a given drug, and I'm not speaking specifically about GvHD, the agencies had to rethink about withdrawing or not the drug.
So, like, if these fail, are these products not available for third line use?
Actually, EMA rules are if you produce a registration trial with negative outcome, you basically out, except which happened in chronic GvHD disease. It was just a totally different line of the provisional approval. Then we could go, so okay, no first line, but maybe third line, because that was the other trials. You know what where I'm referring to.
So the randomized phase 3 trial is a 210 person study. I'm just finding this on clinicaltrials.gov. It's 210 people. It started in late 2021 and is not expected to complete accrual until 2027.
Wow.
It has been extremely slow, Corey, because obviously people have their own recipes. But also, Robert mentioned the compassionate use program. And in many places, you have also some homemade MSCs I'm aware of. So it makes things a little bit more difficult. And this is why the trial has been ongoing very slowly, I think.
Yeah, this is all correct. In addition, there was COVID in 2021. Which slowed down the trial recruitment. And then look at the primary endpoint, overall survival up to 24 months. This also slows down the readout of the trial. But I think recruitment might be completed next year. Okay, I think we have one last discussion point left. What might be the key clinical consideration for clinical practice when treating patients with acute GvHD with MSCs? I think we already heard that the toxicity is quite low. It's quite well tolerated. So we have often the problem that patients with acute GvHD have concomitant infectious diseases and are heavily immunosuppressed. So there should be this makes this type of treatment preferable to other more immunosuppressive drugs, possibly. And then this point that we already discussed about if a certain organ involvement benefits most from MSCs.
But here, Robert, when it comes to clinical considerations, although I think all regulatory agencies and we also as investigators agree with this, that the overall response is the important surrogate marker for a later better outcome. I noticed more and more in different studies that actually, while overall response include PR, those patients who are going really to be cured or at least live long are the CR or VGPR patient. And this is why actually Paul Martin introduced this concept of VGPR a few years ago. I don't know, what is the experience in the group?
For me, I agree. I have the feeling that patients that achieve PR, this is the one that are going to lose the response. And that the aim is to have the CR or VGPR at day 28 or slightly later to be on the safe side.
Yeah, I mean, this is in line also what the prior experience with MSCs showed, right? Those that responded and achieved CR, they have a benefit in survival, right? So their survival was better.
but that applies basically for all interventions, right?
Right. Yes.
Absolutely.
It doesn't surprise any of us, I think.